Hello, everyone. It's a real pleasure to be talking to you today and welcoming you to the A-level biology course. For me, biology really is the most exciting of all the sciences, and I'm hoping that I will be able to show you throughout the 2 years of the course why I believe that to be the case. So biology has been labelled the science of the 21st century, and initially that came from the completion of the Human Genome Project. It was a project that was set about to essentially sequence the entire human genome, and from that, by knowing this sequence, try to identify the causes of particular genetic conditions. And once the genes have been identified, that would open up the possibility of looking for ways to cure it. And the first draft of that human genome sequence was made available around the turn of the century, so around the year 2000. And subsequent to that, we've really sort of started to make inroads into our understanding in that field and identifying genes that are responsible for many different conditions as we go. But then in the last few months, of course, we've all become so aware of the importance of biology. It's really come to the forefront with this pandemic, and our reliance on biologists of many different disciplines has really been highlighted. So there are the virologists helping us to understand the behaviour of the coronaviruses. There are the epidemiologists helping us to understand how the disease spreads around the globe. And then, of course, also immunologists and other scientists working on vaccine development. And later on, I'm also going to explain to you the role that ecologists have got in helping us to look at new and emerging diseases as well. Okay, so what I asked you to do prior to watching this video was really to have a look at page 12 in the biology handbook. And on that page, so that's just should just say that's what we're going to be sort of looking at today. But page 12 looks like this. And what I had asked you to do, I've sent you 7 questions that I wanted you to try to think about whether you could offer any kind of answer to them. But also, in relation to each question, could you identify the Year 12 topic that is going to help you explore in more detail that particular aspect, so that you might maybe this time next year feel confident that you can answer those questions. Okay, so before I move on, just give a little warning really that what we're going to talk about could potentially be upsetting for someone, particularly if you have had any experience in your family in relation to COVID-19. And if that's the case, you might prefer to go to the end of my video, the last few minutes, where I'm just talking about the course structure, and then come back to explore these ideas at some other time. I wouldn't want anyone to be upset from what we're going to talk about today. Okay, so question number 1 was simply, what are coronaviruses? We've got this lovely image here taken by a transmission electron microscope. So, of course, viruses are too small to see with light microscopes due to the resolution that they provide. But this image here helps us to really understand what these viruses are. So they're not cells, and in fact, they're not really living, which we will explore in a minute. But what they are essentially particles of RNA, so genetic material in the form of RNA, packaged up inside a layer of lipid that has been borrowed from the host cell in which the virus developed, so inside of a human cell, it developed and it took the lipid layer away from that cell. And then on the outside are these proteins that are known as the spike proteins, and it's those that give the virus the name coronavirus, because it is thought to look a bit like a crown. Okay, so this is the overall sort of taxonomy for the virus, which is actually now known as SARS-CoV-2. That's its official name, but of course worldwide people just refer to it as coronavirus. But they are actually a whole family of viruses, and so the individual sort of species, if you like, at the sort of species level is this, much in the same way as we have our own classification for humans, down to so our ancestors obviously also within the same family, but then down to species level here. And I mentioned the idea that they're not living, and of course that's something that you will have looked at perhaps in year 7, the criteria that an organism needs to fulfill in order to be classed as a living organism. And it's abbreviated to Mrs. Gren, these 7 criteria. If we just glance at those, the virus is not capable of these things, and for that reason can't be categorized as living. So it can't reproduce on its own. It must have a host cell inside of which it's going to replicate. Okay, so there was a sort of second part to the first question, and that was just to think about the size of a virus in comparison with a human cell and a bacterial cell, and also with DNA. Just a quick reminder, hopefully you've remembered from GCSE, 1 micrometer is 1,000 nanometers. So if we start off thinking about something like an ant at 1 millimeter or 1,000 micrometers, and then a human cell, we're getting down somewhere between 10 to 100 micrometers, depending what sort of cell we're talking about. Bacteria, just quite a bit smaller than that, 1 to 5 micrometers perhaps. And then if we keep going, viruses, we're going down into the units of nanometers. So whereas a bacteria would be somewhere in the region of 1,000 to 5,000 nanometers, we're looking at this sort of whole order of magnitude smaller with the viruses. And coronavirus is actually quite a large virus in comparison to others. In between here, I've included a few other things just to give some context to that. So the masks that are worn by professionals have pores of around 300 nanometers, and so of course a single viral particle potentially could pass through, but usually they're within a droplet, and it's for that reason that they're stopped from getting through the pores. And then just to finish this off, so antibodies are fairly large proteins, down around 15 nanometers, and DNA even smaller. Good. Right. Now this is another lovely image. This one came from a scanning electron microscope. So you get this sort of 3-dimensional aspect to it. And the colours added afterwards, it's false colour, but the pink shows us these viral particles budding out, sort of emerging out from inside of this host cell. And so, as I mentioned this already, but we can think of the virus as being this sort of packet, if you like, RNA enclosed by a layer of lipid that has been taken from the cell in which it formed, and then on the surface, these spike proteins that have a particular role, which we'll see in a minute. So how did you get on? Let's see. You can write to tell me if you'd like. But these are the topics that we have touched upon so far. So cells, we look at electron microscopes, their uses, and also think about the relative sizes of organelles and different types of cells. We've mentioned RNA, which is an example of a nucleic acid. So we're going to be in topic 1.4, comparing RNA with DNA. And we've also mentioned a little bit about classification, which comes up in 1.6. So question 2 was, how do viruses replicate? And that's, again, where we need to think more about these spike proteins. So these are these large sort of proteins on the surface that make the virus look like almost like a crown. And these spike proteins are able to bind specifically with proteins in the human cells, on the surface of the human cells, that are known as ACE2 proteins. Now, once that has happened, that interaction, the viral particle can sort of basically unpack its contents inside of the human cell. And so the genetic material can then give instructions for formation of new virus. So we've said that the virus contains some structural proteins like these spike proteins. It also contains a number of enzymes that are going to help to control this process of protein synthesis, making more viral proteins. So by making more enzymes and making more structural proteins, all of the requirements are manufactured within the cell here in order to make new virus particles, and they're then packaged up. They take some of this membrane away from the host cell, and then these new virus particles are released, much like we saw in that nice scanning micrograph image. Okay, so that question looks at the role of enzymes, which we do in topic 1.2, but also protein synthesis, that we're going to explore in much more detail than what you have done at GCSE. Question 3 now was simply, how does washing your hands with soap and water help to inactivate the coronavirus? And so to answer that one, we firstly need to just think for a minute about what soap does in general. So soap is an unusual sort of molecule. It has two kind of distinct regions to the chemical molecule. And the sort of one region is referred to as being polar, and it will interact with water. And then the sort of what's often known as the sort of tail region will not interact well with water. And so then when soap particles bind with dirt molecules on the surface of our skin, the non-polar part will interact with that dirt, and the head region will face out towards where there is water. And in doing so, that helps to lift the dirt particle away from the skin and off with the water, so it gets carried off as we wash. So in a similar way, we've already said that the virus contains this layer of lipid, and these soap molecules with these two different regions, the head region is correctly referred to as being hydrophilic. It likes to interact with water, and the tail region is hydrophobic because it avoids water and will therefore interact with lipid oil. So the tail region will interact with the lipid within the virus packet and help to sort of break that viral particle open, that sort of lipid layer open. And then as the particle breaks apart and all the components break apart, they're each individually surrounded by soap particles. And then the actual process of the water, the movement of the water, helps to carry them off. So that's why washing our hands for long enough with soap and water is a really effective solution to destroy the virus. Of course, if we just leave other surfaces for long enough, then the virus will also disintegrate. But there's been quite a lot of studies done about exactly how much time we need to leave different surfaces for to become decontaminated. And so cleaning is obviously a way of helping to speed that up. Okay, so that question looked at the role of water and also what are known as phospholipids, which are these particular lipids we find in membranes. And so that is in topic 1.1. We also look in more detail at the structure of the cell surface membrane of our own body cells in topic 2.3 as well. On to question 4. Now, this was the one that probably leapt off the page. Certainly when I started to plan this was the one that was most immediately obvious. So how does the immune system respond to the virus? No prizes for working out that the topic here is 2.4, the immune response. But so how does the immune system respond? So the viral particle, once it replicates, becomes in increased numbers within the body, and these viral particles then get taken up by particular cells of our immune system and presented. It's a way of sort of flagging up to the immune system that this virus is here and that a response needs to occur. And one of the first cells to respond will be a type of lymphocyte, type of white blood cell known as a T helper cell. And once it has this specific recognition to the virus, it becomes activated and it starts to release some chemicals known as cytokines. So we could literally translate cytokines as chemicals that cause cell movement. And so these T helper cells release these chemicals and help to activate other cells of the immune system to also respond. So without the T helper cell, none of the other things will happen. But once they are active and releasing cytokines, this will help to activate B cells that make antibodies, and also another type of cell known as a cytotoxic T cell. So cytotoxic cell killing, if you like. And what they do is destroy any of our own body cells that are infected with virus. And once they do that, the virus is free and it makes it visible for the other cells to start to destroy. So obviously some of the negative symptoms that we might experience from this infection, or from indeed many other types of viral infection, come from the fact that our own body cells are getting destroyed. But in the case of COVID-19, there also seems to be situations where it gets more serious, where there seems to be something which is known as a cytokine storm, where essentially too much cytokine is being released by these T cells and it's triggering too much of an immune response. Our immune system is obviously really vital in protecting us, but if it goes into overdrive, it can actually be very dangerous for us as well, for our own body. And that seems to be one of the key problems that has occurred with some patients. Okay, now, so if we just think for a minute about one of the main kind of systems of the body that's affected by the virus, we know that our own alveoli, if they're healthy and we haven't got an infection, are the sort of site of gases exchange with this increased surface area, or the alveolar surface here with the capillary surrounding it. when the lungs become infected, depending on the extent to which the infection takes hold, if we start to get this increased inflammatory response, there can be some build-up of fluid, and that can cause the sort of coughing and the breathlessness and the other things that people are reporting as a consequence. Now, question 5 also links to the same topic about the immune system, so I thought we'd deal with that one at the same time. So the whole goal with a vaccine is to find a suitable composition to a vaccine with antigen that's going to be triggering formation of really specific antibodies that will help to target these, perhaps like these spike proteins on the surface of the virus. So if a person has already had a vaccine with, say, for another illness, so for example, like a flu vaccine, these antibodies are not going to be specific in shape and therefore not able to interact with the same proteins on the coronavirus, and so they don't offer any protection. But if we can find the right composition with the right antigens and other components to trigger a really specific formation of an antibody that's protective, then that will be an effective vaccine, because what will happen is the B cells in the body will start to make these antibodies, which will then bind in this sort of specific interaction. And if you remember, we looked earlier at what the spike protein does. This protein on the surface helps to bind to the person's cells and helps the virus to enter the person's cells. So if that's blocked, it means that, of course, that can't happen, the infection can't take hold. And then from there, it's just down to the other cells of the immune system to clear up the virus that already exists. But if it can't get in the cell, it can't replicate into big numbers, and then the person is safe. I'm not going to explore this now, but you might want to be thinking about, as an extension, how will the scientists who are developing vaccines, and there are many different groups around the world all trying to work on this as we speak, but what are they going to need to do, and how are they going to evaluate whether their vaccine is effective? So you could think about, from GCC, the work that you did about drug development, that would be relevant here, and how trials are done. Okay, so obviously that topic that we've just talked about is mostly the 2.4, the immunity topic, but when we were thinking about gases exchange, that comes up in topic 1.3, as does looking at the exchange in the digestive system as well. Right. Question 6, how can we predict the risk of another emerging zoonotic infectious disease in the future? Now, I've heard a very interesting podcast by Professor Kate Jones from UCL about this, and that's why I decided to include this for you. So, firstly, what is a zoonotic infectious disease? Well, it's a disease that can be, or a pathogen that can be passed from animals to humans. And what I should say, these coronaviruses, from that family of viruses, the one we're talking about currently that causes COVID-19 is not the first to have been able to transmit to humans and cause disease in humans. So we had SARS back in about 2009, and we also had another one called MERS in between. But they didn't quite cause the same extent of problems as the current one has. Now, what Kate Jones was saying really was that she's identified that as humans increase in population, we're having to intensify our use of the available land for agricultural purposes and just for housing and so on. But that's then changing the interactions that occur between humans and other animal species. So contacts are happening that may not have happened previously. And the other thing is, of course, that we move around the planet a lot more. We move livestock a lot more readily than we would have done in the past. And so it's estimated that up to somewhere around 3/4 of all human infections are either as a result of a zoonosis, so a direct transmission between animal species to humans, or there's some sort of vector involved as well. So, for example, like with malaria, with the mosquito as the vector for that. So, hopefully, what this illustrates to you in a very whistle-stop fashion is the importance of ecology in all of this as well, because by carrying out ecology studies to identify the numbers of different pathogens in various animal species and also the diversity of these pathogens, it's going to provide us with more information for the future in terms of modelling where and perhaps even when further zoonotic diseases are going to emerge. And of course, having that knowledge means that we can then actually try to put in place preventative measures to stop it from happening. So that idea there, looking at ecology and looking at diversity, comes up in 1.7, the biodiversity topic, right at the end of year 12. There was one final question for you, and that was how do we test for a coronavirus infection? And the really short answer is that you're going to have to wait until you're in year 13 to find out in detail when we do the DNA technology topic. But if we just briefly consider, you may well have even seen this on the news, a piece of kit like this is being used, which is known generally as a PCR machine. And so PCR is a process of essentially copying genetic material many times. It requires enzymes to help control that to happen, and it requires more nucleotides to build the building blocks for the genetic material. But once those things are in place, the test can be done in such a way as to try to detect very specific sequences within the genetic material that have been identified as belonging to this particular virus. This just summarises that process of firstly making more copies of the genetic material for the PCR process. So it's a bit like genetic photocopying in a way. And then once that has been done, using labels to very specifically identify particular sequences. So if any of this is really sort of taking your interest at all, I'm aware that you've got quite a long time still in between now and when I get to see you in September. And so hopefully you might be able to explore some more of this for yourselves. And I can send these links out to you via email. Perhaps it'd be easier for you to have a look at them that way. But the Imperial College are doing a whole series of what they call lockdown lessons on YouTube at very specifically advertised times. And some of them sound really fascinating. I'm hoping I'll be able to catch some of them myself, actually. But do have a look. They started in mid-June, but there's quite a long list of them spreading throughout June and perhaps even into July, so do have a look at those. And then the Exploratorium, often that's designed for younger children, but some of the activities they've got on there sort of help to inspire some of what I have talked to you about today. And they've actually got some suggestions for little practical activities you could do at home to think more about the lipid coat for the virus and how it behaves and that sort of thing. And then there are the podcasts that I mentioned as well. Now, in just very briefly, I want to just go over some of the sort of day-to-day about the A-level course, in case you're interested. This is all summarised in the handbook anyway, but just in case. So you have a total of 4 hours of biology on your timetable each week, and then you are expected to do at least another 4 of independent study on top of that. But with the 4 hours in lessons, you have 2 different teachers, so 2 hours with each. Things you should bring with you to lessons: pencil for diagrams and graphs, a ruler also for graphs, calculator because we do quite a lot of maths, lab coats when we're doing practical work. It's a good idea to have a copy of the textbook, and I've got the ISBN numbers for you in the handbook. And then on top of that, you must have some way of keeping your work secure. So it should be in a ring binder, not just sort of floating about, and you should have it divided up into the different topics. As with GCSE, we hand out work packs for each topic, and there are tasks in there that relate directly to what we do in lessons, so it's important to have those with you as well. Right, now, because A-level is a step towards much more independent learning, I just wanted to highlight to you the importance of this. What you're doing at A-level is really building up a whole series of new knowledge and understanding. And once you have that, we need to actually learn how to apply it to new situations, be able to analyse data in relation to those ideas, and also do things like evaluate other people's conclusions. Now, these are the skills that are definitive of A-level, and the students achieving the highest grades are the ones that can really confidently carry out these skills right at the top, as well as all of this at the base. And so what you can do to help yourself with the course is really to read ahead of lessons to ensure that you're building your understanding sort of consistently, and keep going back over things, keep testing yourself with little recall quizzes, either with friends or with family members, to make sure that you're building that knowledge up all the time. And then that frees up more of our time in lessons to help you with some of these other higher level skills as well. Now, there are 6 practicals that you're required to do in Year 12, and then another 6 that you're required to do in Year 13. We do many more than that, but these are the sort of ones that are stipulated by the exam board. So very briefly, that's what they are. And you're provided with a lab book where you keep records of the work that you do for these, and I can explain more about that once we get to that stage, because we need to plan them quite carefully and really be able to explain why we're doing the steps we're doing and that sort of thing. And that goes towards what's called the practical endorsement, which is an additional qualification that sits alongside the grade that you achieve for your exams. So you sit your exams, and that determines whether you get an A or an A star or whatever. But then alongside that, there's also a recommendation whether you get a pass for what's known as the practical endorsement. And really that depends on the evidence that you build up in your practical work, and also the level of independence that you start to be able to demonstrate with that. And then, so lastly, that's the textbook. There are 2 sort of smaller textbooks, one for each year. or there is one larger one. I think you may save something overall by purchasing just the one book, but of course it's heavier to carry around, so it's really down to personal preference. Okay, so that's it from me for now. I hope that that's given you a flavour at least of what we're going to be doing throughout next year, and I'm looking forward to exploring all of those ideas I mentioned in far more detail with you from September onwards. But in the meantime, make sure you take care of yourselves and make sure you have a good summer holiday. And that's it from me. Thank you.